In my Genetics 311 mini video, I'll be following the 3.2b prompt illustrating the key findings and experiments by Nirenberg and Mattaei and Nirenberg and Leader. So Nirenberg and Mattaei were the first to discover that a specific RNA sequence coded for a specific amino acid. This discovery was made in 1961 through an experiment where they wanted to determine what amino acids are specified by a codon of a single base, such as U or AAA. So their experiment will be demonstrated here through my at-home model. So Nirenberg and Mattaei determined that if they added a single base mRNA strand, otherwise known as a homopolymer, which will be represented by this, to a cellular matrix with the 20 amino acids, then the resulting protein's composition would display what amino acid the single base mRNA coded for, because it will be made by the amino acids coded by this mRNA strand. But since there are 20 different amino acids, the experiment had to be performed 20 different times, with different amino acids being radioactively marked with C14 each time. This will represent a radioactively marked amino acid. Now, if the protein produced was radioactive, then the marked amino acid for that experiment could be linked to the single base mRNA strand. So the first homopolymer they tested was poly-U, or triple U. They added that to the cellular matrix along with the 19 amino acids, and the one they radioactively marked was phenylalanine. And what would you know? The resulting protein was radioactive, allowing them to conclude that the codon, triple U, the poly-U that they used, coded for phenylalanine. They used the same method to determine that triple C codes for proline and triple A codes for lysine. Nirenberg and Leader performed a similar experiment to determine the matches between the other amino acids and codons that weren't only homopolymers. This will also be represented by my at-home model. To do this, they added short mRNAs with known codons to a mixture of tRNAs, with their attached amino acids, put that in there, and ribosomes, right here. And the ribosome would then bind the mRNA to the appropriate tRNA, like such, and then the mixture would be filtered out, leaving only the tRNA paired with the ribosome-bound codon. But just like last time, though, in order to determine what amino acid was attached to the bound tRNA, it had to be radioactively marked. So for each experiment, one amino acid on a tRNA was radioactively marked. If after filtration, the tRNA bound to the codon was radioactive, such as this, then it can be concluded that that specific codon coded for that specific amino acid. This procedure was used by Nirenberg and Leader to identify 61 different codons.